Hi, my name is Victoria Perello, and I participated in the summer undergraduate research experience doing research with Dr. Sergey Yakovenko in the Department of Human Performance Exercise Physiology. My research compares the abilities of rate models and spiking neural networks to handle noise when simulating a biological phenomenon called central pattern generators. Neural networks are algorithms that mimic brain connections and simulate neurological functions using connection weights and nodes. Spiking neural networks are a type of artificial neural network, but they differ in some key areas. Traditional artificial networks use static or dynamic inputs through a series of layers that are then backpropagated to fix errors in the connections. Backpropagation is a method of minimizing errors between actual and expected outputs to optimize connection weights within the algorithm to improve results. Spiking networks use spiking in neuron inputs and recurrent connections instead of backpropagation techniques to output an approximation of the results. Central pattern generators are biological neural circuits that control the rhythms generated during locomotion. This biological phenomenon can be shown by using a system of ordinary differential equations of half-center oscillators, which are then solved in the models. It is very important for biological algorithms to be able to handle noisy data due to the nature of biological data. Biological data can be noisy for a multitude of reasons: interference from other sources, bad collection techniques, and poor instrumentation. The goal of this research is to compare these models' ability to adapt to noise and data. My hypothesis was that when comparing rate models and spiking neural nets with noisy data, the spiking network would be more adaptable and process the data with biologically plausible results, whereas the rate model wouldn't converge or would fail to be biologically plausible. In order to test the theory that spiking neural networks are better at processing noisy data than the rate model, I needed to add noise to biological models. I used a rate model of the bilateral CPG in MATLAB and a spiking model of the bilateral CPG in Nango, a Python package for creating spiking neural networks. For both models, I added noise using a Gaussian distribution function. These functions chose random numbers between 0 and the standard deviation that I selected. Seeing as the models operate as integrators and oscillate between 0 and 1, I determined that by choosing a standard deviation starting at 0.01 and increasing by that same interval, I would be adding a 1% change in the variance of data. The results of this experiment were compared to the models run without any noise added to them, showing optimal model results. As we can see in the data shown in the following figures, the rate model on the top, which shows a very clear gate of swing and stance phases in the result with no noise. This biological gate is not retained when noise is added. We can see multiple instances of both swing and stance phases multiple times in a row, which is not biologically possible. In comparison, we can see in the spiking model that while the phases are not as linear in the beginning, they very quickly linearize. This is caused by the connection weights optimizing throughout the simulation. In fact, in the results, the spiking neural network with 9% noise, we can see that the connection weights take almost 25 seconds to optimize, but the model eventually picks up the model behavior in a biologically possible way, confirming my hypothesis. This process of adding noise to these models is novel because I have not found any evidence of this type of comparison being done in any of my research. This comparison shows the model's ability to process real biological data, not just optimized input data. This research can be used for future clinical simulations when doing research on different types of injuries and diseases. For instance, a simulation could be used to show the impact of a neurodegenerative disease like ALS and show that the body will respond when different parameters are changed. This could show how both rehabilitation and patient care could be conducted. Based on this research, we can conclude that spiking neural networks do in fact process data with noise and maintain biologically possible results, which is not true with the rate model. This is very significant in the realm of computational neuroscience, where one goal of research is to improve techniques of simulating complicated dynamic systems while reducing computational cost. Computational cost is reduced by using less complicated models that make assumptions about systems and lose accuracy. These spiking models show that we can improve accuracy and include more dynamics in our models by changing the amount and the type of inputs we use in neural networks. I hope to continue research similar to this, using spiking neural networks to simulate other biological phenomena and finding a way to simulate neurodegenerative diseases.